find something.
My job is changing where I get to start chasing more funding and maybe trying to chase funding that'll leverage iPlant resources.
Got it. So you're looking to get the overview half so that you can help leverage it for pretty advanced topics, which you're probably not going to get out in today's workshop.
Right.
If you're doing
that goes all
but-
alone, and I'm, at the same time as I'm doing this, I'm diving into the resources that you guys are suggesting.
Yeah, so you can also figure out how to sell this if you prefer going out and chasing some more funding.
Yeah.
That's fantastic.
I want to sell it to more people using it.
Perfect.
More users.
We like sales reps. That's fine. We'll give you great commission too.
Password does not
We
work.
read the letters of support
Okay.
all the time.
So
Snickers bars.
Yeah.
I keep trying to get peanut
you have to
M&Ms.
have, there's
How
some
would you-
obviously.
I'm Higgin. I also work
special
for
business.
Dave, and I'm a software developer, and I'm kind of curious. There were some, I guess, workshops on the HPC stuff, and I want to get into it. I said this was sort of pre-rec.
You know, teaching the HPC section?
No, Rob.
Susan. Yeah, so
It's all just in my office.
that'd be really funny. It's like the only way you're going to learn anything from the person next to you is to come over here, which
These are
is
the only ones.
across the hallway.
If you can remember that.
Neutral territory. I could understand that. So we have 3 people that will identify as software developers and probably familiar with the command line. Good. They don't nod much.
They typed it in their mind. My name is Blake Joyce. I am a bench scientist that dabbles
Yes.
in the command line. So that's, I guess I'm sitting geographically in an accurate place. But my background is kind of varied. I come from an ecology background, but I've done a lot of plant biotech. We were actually responsible, Neil Stewart's group was responsible for the extremophytes for the KP project. And I personally harvested 20 of those, which required a lot of travel. So it's nice to see that showing up in other places that actually is getting used and real. That's great.
Thanks to them for that publication.
Yeah, well, Gain and all those guys are looking for something big, so I don't know how long it's going to take to
The
go on.
longer you shop it, the lower
Yeah,
impact factor
yeah,
it
there's
becomes.
the shelf life is what you're talking about. Yeah, absolutely.
Old data stinks.
Yes, it generally does. And so I'm here to just kind of learn more of these things that are user interface type things.
Looks good.
I grew up in the beginning of assemblers, and they've changed a lot. Can you get my short time? So I need to get up to be. Right now I'm in market for that, but we'll be doing that.
Okay, you could totally change that, no problem. So
Get back in the
an
game.
email to validate.
Cool.
And then should be good.
I'm quick. Just get transferred from bench work to purely theoretical work. I'm working on modeling, ground simulations, DNA sequence. I guess I will should learn. Postdoc in my lab says this is a really good chance to learn something, so that's why I'm here.
Okay, so if you're developing models, you may be really interested in terms of how you can leverage a lot of the large-scale distributed computing, especially if you're trying to test a whole bunch of
Yeah,
things,
yeah.
parameters, sweeps, or different starting points, or whatever it is you're simulating.
What kind of models?
Right now my project is simulating the evolution of DNA sequence.
Cool. A's change to T's.
And sometimes back.
That's back.
I'm Laurel Johnstone. I'm a research specialist at the genetics core, and I was trained in biology, but past few years I pretty much just do bioinformatics stuff. And lately I've been mostly working with human exome sequencing, and I have my own pipelines for analyzing that data on the HPC. And I kind of want to see if, I could do something similar through the discovery environment or atmosphere or something.
Yeah.
And whether that would make it easier to, collaborate with team members or track changes to pipelines, things like that.
I would say if you've got pipelines that are requiring those kinds of resources, it would be great to see them integrated on iPlant infrastructure, just so that hopefully it makes your life easier in terms of running and then sharing the data afterwards, but also that other people could
Other
get
people
a
can
one-
run it for me too.
can run
would
it
be
for
nice.
you, exactly. Really simplify that process. Because one of the things that often happens, especially as you become known for being able to do a particular kind of analysis or set of analyses, is people just come to you. And the scalability isn't necessarily the compute resources you have, but the amount of time that you have per day to run and monitor and take a look at these jobs. So it's nice when you can scale that horizontally to other people to help with that. Better yet, empower people to do it themselves on your stuff, and then they can go train the next person after that. That's cool.
Okay, I'm Hui. I'm from Department of Pharmacology and Toxicology. I'm a graduate student, and my lab is working on liver disease. And it seems like
Can you speak up a bit?
I'm sorry. So my lab is working on liver disease, and so in my research, sometimes we'll generate some data from the metabolomics or from DNA sequence, next generation sequence, something like that. We know nothing about data.
Okay.
Interesting. Interesting. Do you have any experience on the command line? and how
No,
to
so I
program?
have zero experiments on bioinformatics, so I have nothing to do with my data. Instead of just send them to our collaborators and
Got it.
let them to
Yeah.
process that. So that's why I'm here.
So this is great, and it's interesting that you guys have almost self-arranged a particular gradient from the professionals on the computer to the professionals in the lab. And we can do a couple of rearrangements here to get people lined up. But this does sort of represent a lot of the typical audiences that we have at these kinds of workshops, which represents an opportunity and a challenge at the same time. And what it is these resources that iPlant has to offer really tries to provide tools and services and the knowledge on how to use them to both the professional programmer as well as a professional biologist. And in essence, it's bringing your two brains together where we really can have some unique discoveries happen. And hopefully iPlant's infrastructure helps lower the barrier to make those kinds of conversations occur. So, but core to iPlant, and one of the things I think is really important to remind people is that biology as a whole is entering into a really unique era. And it's fun to sort of be part of a revolution. And out of curiosity, do you guys feel that you're part of a revolution? Do you hear the call to arms? Are you taking up those pitchforks of pure data and charging forth into uncharted territories?
Yes, absolutely.
I hear a call to arms. How about the rest of you? You look very quiet.
Yeah.
I concur.
So just look at the questions that were
I have-
asked 10 years ago.
I just want you to know that I'm up here for another hour and 15 minutes, and I look for entertainment from all of you. So I never really want to forget the wonders of biology. And just to shut off these lights and hopefully just take a look at this picture a little bit better. One of the things, hopefully, even for the most hardcore computer science, have you spent some time outside? I remember when I used to work at a software company, we'd always call it the Great Blue Room. Where are you going? I'm going to the Blue Room. Oh my God, be careful.
People out there.
We don't know what's going to happen to you. But all I know is don't stare at the bright thing for too long. It doesn't seem to do well on the optics. Have you been outside a little bit?
Yes, occasionally I get some sunlight now.
Good,
But-
good.
Vitamin K, yeah.
I'm going to assume that at least you could recognize these things as flowers.
A little weird.
A little weird looking. Anyone here a plant biologist by training? You know what these flowers are?
I don't know the genus species for those, but I think they're an orchid, right?
No, they're in the order of ABLEs. These are little tiny. You ever see those white panicles of flowers? You ever bend down and smell them, and they're sort of sickly sweet? Like they sort of smell good, and they sort of smell like something's rotting at the same time. Do you have any idea what I'm talking about?
Yeah, yeah.
Okay, good. Anyone else see flowers? I'll come over to this
Really
side.
small.
Little tiny, small flowers. If you bend down to smell them, they're sickly sweet. And they're characteristic of what we call a pollination syndrome, which is a particular kind of morphology, phenotype that an organism a flower is showing in order to attract a particular kind of pollinator. So flowers in this order, ABLEs, are characteristic of being pollinated by flies and bees. The reason I'm telling you this story is one of the great things about biology is that there are all these fascinating stories to tell. And if you're the plant biologist, you may focus in on the flowers. Anybody here an entomologist? Anyone here know what entomology is?
I'm married to one.
Study of insects. Yeah, we're going to have to bounce it around, and I'm very happy when I ask biology. You'll get an odd. We'll switch over to computer science later. You guys will be pros. Perfect. Study of insects. Can you recognize this insect for me?
Cicada?
Close.
It's a fly, isn't it?
It's a fly.
That was a trick question.
The trick question. Just a nice fly right there. So and this is not just any fly. This turns out to be a very special kind of fly.
Dead fly.
A what?
A dead fly or
A
about to
dead
be
fly. You obviously see the next stage. So, did you see that at first? Doesn't matter. We'll come back and test you later. So this is a flesh fly.
Oh,
These are the flies that the females go around looking for dead, rotting, decomposing meat in which to lay their eggs so they're developing children, have a delicious nutritive environment in which to grow up. But it doesn't look like this one's going to get a chance to lay too many eggs because who said it was a dead fly? It's a dead fly. Tell me some more of the story.
There's a big spider.
There is what appears to be a gigantic spider here. Do you know what kind of spider that is?
I don't know. White
It's a-
spider?
It's a white spider. It's a crab spider. Did you not see it at first?
Now she's got the heebie-jeebies. Way to go.
So just to put this,
Computer science, it's gross.
yeah, put this in, did you not see it at the very beginning? Isn't that cool? Isn't that really cool? Do you know what that's called when an organism is hidden from view? Camouflage, also known as, what kind of coloring is this? Not mimicry.
oh.
Cryptography is sort of related to this, hidden. Cryptocoloring. So this is a cryptically colored organism. So, I'll come over and pick on the computer science. No, I'll come over and pick on the biologist. On the very end, Hui is your name, right?
Yeah,
Why is this thing cryptically colored?
protect himself or herself. I don't know.
This is definitely probably a female. Females tend to be much larger in the spider world than
Oh,
males.
all right.
Protect from what?
I don't know. Dangerous.
This is cryptically colored to protect from danger. What are you afraid of? Why would you be cryptically colored and hiding from danger? Told you I was going to pick on the biologist. Put yourself in the mindset of any organism. Why do you want to hide? Why do you want to hide? I see you hiding behind your computer.
Yeah.
Why would you punch a bear?
Why would you not punch a bear?
Yeah, just for attacking.
That's a perfect one, exactly. So one reason why this organism may be cryptically colored is to hide so that they can attack, bite, inject full of a delicious cocktail of neurotoxins and digestive enzymes an unsuspecting food item so that it then can go ahead and have a tasty meal. However, in this case, it's not correct. You see these dark markings on its abdomen right here? These turn out to be highly reflective in the UV spectrum. And what that means is that these are visible and actually serve as attractants to flies and bees. So that flowers that have these spiders on them are visited much more frequently than those without. And the reason it's cryptically colored really comes close to that idea that you had, Blake, which is, this is the same reason why we don't run up and punch a bear. Which is they want to hide from their own predators. And a lot of those can be birds and mammals, other vertebrates and things along those lines there. In this case, when I took this picture, I was actually focusing on the fly. I really wanted a picture of a flesh fly, and I didn't even see the picture of the crab spider. Put it into perspective, like this entire crazy scene here is operating on a scale of about 1 centimeter. So it's not nearly as impressive as when you see it up here. And I didn't even notice this spider until I had a chance to take a look at it on a big screen. And this is what I love about biology. We've got so much neat stories to tell. And of course, we've got some cooler stuff too, not just static pictures, but we have things like behavior. Computer scientists, what are these? Birds. These are birds of paradise from Papua New Guinea. You, which one is the male?
That one.
That one right there.
The one scares the other.
Yeah, obviously.
What's he doing?
Trying to get his attention.
Trying to, was he successful? No.
Very hard to tell. That's David
I mean,
bro.
this is incredible. You take these static things and you give them this stuff called behavior, a little bit of selection, and you get some incredible outcomes, both in terms of their performance, but also in terms of their morphology, sexual selection. Incredible things to witness in all animal species, spiders included, it turns out. Very, very neat stuff that goes on there. What I want to emphasize here, that there are more stories to tell. And this happens, of course, down at the molecular level. This is something that's often hidden to us from many, many, many stages. And this is an incredible animation that I had nothing to do with. This is Harvard's BioVisions. And what they're doing is showing the molecular processes of a white blood cell, a leukocyte, identifying a site of inflammation and triggering a set of communications to transfer information from outside of the cell inside of the cell. And what I love about this is it puts to life, in the same way that we can see the behavior of organisms, the behavior of what's happening inside of cells. And I don't know about you, but when I was taught biology, what I was taught is that these things are essentially bags of goo. You just have a bunch of stuff crammed in there, it's sort of wet, and really neat stuff happens that gives rise somehow to life. And in reality, what we're dealing with are these highly dynamic molecular regimes. And what we're looking at in terms of these tables are the intracellular structures and fibers that give cells their shape in terms of automatically assembling and deassembling. But these things are also used as the intracellular transit system. This is one of my favorite proteins if you've never seen one. This is kinesin, thiosin. These basically are motorized walkers. They're burning ATP and moving those little feet. And they're traveling along these microtubules, which basically give cells their structures, and they'll drag stuff. So that one was dragging a vesicle filled with something from one part of the cell. Oh, goo.
And that's not real speed either. Some of them go as fast as 60 miles an hour.
Oh yeah, they're broken. Yeah, and in terms of animation, just to give you a perspective of how awesome it is inside of the cell, when they started to try to model this and make this animation using real data from real scientists generated over tens of years, 20s of years, hundreds and hundreds of labs, is that they realized that in order to really make this animation look good, they had to expand the intracellular space by tenfold in order to get their virtual camera flying around there appropriately. Just phenomenal stuff that happens inside of the cell. And so what I just want to emphasize here is that as we think about all the beautiful ways that life operates, all of this comes down to an instruction set that is encoded inside of what? Testing you guys here. Those guys, who said that they were in the, what facility were you in?
The genetics course.
Genetics core. What's that genetic stuff? DNA. What do we call all the DNA when it's assembled together?
Genome.
Genome. All of this comes down to a genome. What does that genome look like?
Well, that, or did you mean chromosome structure?
This is how we represent data. This is how we represent the data that gives rise to all that beautiful meaning of life.
Yeah.
And there are very few people out here. What's your name again?
Hagen.
Hagen. Hagen is one of the few people, based on his intimate knowledge of biology and years of studying it, where he can take a look at something like this and say, you know what, out of this gigantic pseudo-random strings of A, T, C's, and G's, I can see T, A, C, A, G, A, 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 T, T, G, D, G, 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 T. I know exactly what that organism is. And I would look at Hagen and I'd be like, I don't believe you. Hagen would look at me and say, that's E. coli. And I'm like, you're a genius. How do you know that? It's like, I just know these things. And in essence, what we deal with is sort of this challenge that's happening in biology. And for those that are coming out of ARL, you understand more than a lot of people what's happening in terms of data, which is the incredible growth that we have in front of us. And so this is a graph that when I was an undergraduate and a graduate student, I would track. And I'd look at this every month because I wanted to know how much more sequence data have we as an entire planet generated. So this started back in 1982. And what this basically shows is that over time we were seeing this exponential increase in the amount of available DNA sequence information. And I would track and track and track, and I love this plot. I would use it in talks. And then suddenly, in 2008, they stopped updating this figure. And you can still go to this website, and this figure is still there, and they are no longer updating it.
Because there's so much that they can't keep up.
Exactly, what happened?
Next generation sequencing.
Next generation sequencing. So this is another graph by NHGRI that really shows this change that is happening. Again, I'm just using DNA sequencing as an example of this. It's happening across all of biology, which is the explosion of our ability to get quantitative measurements about the world around us. So cost of DNA sequencing, 1 megabase, for those that don't understand biological terms, what this means is 1 million nucleotides. How many of those A, T, Cs, and Gs we can sequence, and how much does that cost? So this would track Moore's law for many, many, many years. For those that are not computer scientists, the computer scientists can tell you easily, without any hesitation, look you straight in the eye and tell you what Moore's law is. Isn't that right, Hagen?
Yes.
Can you look at Blake and tell him straight to the eye?
Maybe not straight in the eye, but I can say.
What is Moore's law?
That the progression of technology is, well, at least for like computer processors, you can produce faster and faster machines over the course of a period. And it's set at a rate, and I don't remember what the rate specifically is.
It's exponential, yeah. So basically what happens is that Moore basically made the observation that every 18 months, the number of transistors that we're able to fit into a microprocessor is doubling. And we've taken that to interpret all sorts of things. The speed of processors is doubling every 18 months. The rate at which sequencing is, we're able to generate it, basically doubles every month for the amount that we can pay for it, or the cost halves. But something happened right here. And you said it spot on. What we call next generation sequencing. What this was a new technology that allowed scientists to do massively parallelized DNA sequencing simultaneously. And the cost just plummeted, super exponential. And when that happened, our National Center for Tracking Biological Information said, we give up. More data is coming, and it's so cheap it makes no sense to actually track it. And this is a culprit, this machine here. This is a machine that you have seen, and you have seen, because it is in the wet part of your lab. This is a HiSeq 2000, upgraded to a HiSeq 2500, soon to be a HiSeq Infinity. But basically what this machine does is in 11 days, it generates about 4 terabytes of raw data, which get processed into 600 billion bases of DNA sequence. And that equates to about 200 human genomes. To put that much information in perspective, what I want to do is reflect back on the first human genome project, which generated about the same amount of data. First human genome was released in 2000, 2001. It took hundreds of people in dozens of labs located all around the planet to generate this much data. It took, I don't know if I said this, tens of years, 10 years, to the cost of $1 to $3 billion. Today we can generate about the same amount of data in 11 days for a few $1,000s, maybe $10,000 to get a whole machine to yourself for those 2 weeks. So what this has done is it's really liberated high throughput data acquisition. And it leaves us with this next question of how do we and what do we do with all of these data? So what is a human genome size? How big is a human genome?
Small compared to a plant.
Small compared to, it depends on the plant.
Yeah, that's true.
Definitely.
What do we got? 6.5 billion nucleotides, so half
Oh,
of that.
you are the first person to basically say, you know, this real size of a human genome includes both parents.
Yeah.
It's 6 billion bases. Most people say it's 3 billion bases or so. To put that into perspective, you know that you get chromosomes? Half are from mom, half are from dad. If this is a mysterious process, We can cover, the internet has a wealth of information. We can start that now.
That it does.
And just for like, parlor tricks that you can talk at home, if anyone ever asks you how big a human genome is, if you were to take, one set of those chromosomes and lay it out end to end from a single cell, it's about 2 meters tall, 6 feet, thereabouts. That's how big a human genome is. But this is not just limited to DNA sequencing technology. It's happening everywhere. And ecology is a really fun place where this is happening. You are an ecologist, and you said something about ecology that you like, which is you get to travel.
Oh, yeah.
Yeah. So for your friends or your family, this is usually what it means, which is, hey guys, guess what? We're going to
Don't
hang
get
out
mad
on
at me,
Netflix.
yeah, absolutely. I've actually done that in the southwest of Georgia.
Yeah, exactly. We're going to go dig somewhere you've never digged. Ecologists are awesome to hang out with. They will take the most crazy, desolate spot and try to make it interesting for you, just so that they have company. So if you're lucky, you end up over here. If you're unlucky, you're over here. And then what happens is they'll take very detailed notes about what they've seen. For example, I saw a fish, and here's a lot of information about the fish. And if you're less lucky, you might not be able to actually read what they're writing down in their notes. This is not what we call scalable data recording. But we do have a whole bunch of new technology that allows us to track and understand our natural world in ways that are completely unprecedented. And this is one of the things that I think is fascinating. So what people are doing is they're going out there on boats and they're catching great white sharks. Because, hey, why not? It's easy to catch a great white. Haul it on board the ship and attach to it a bunch of interesting sensors on the back there that basically allow them to track the position of the shark, where it is within the water column, and a whole bunch of interesting physical care. Are you okay looking at sharks?
Yeah.
Great whites, no problem. Little spiders freak you out?
Yes.
Man.
They will eat your ears.
I would say absolutely true. And so this is just the neat thing about technology. Like, relatively speaking, this stuff is cheap and inexpensive. And what you can do is now go out there and hook up as many sharks as you can, have them relay data to satellites or other kinds of ground-based towers, but then relay that to a central computer that I can log onto a website at any point of the day and check out the really important questions of life. Where are the great whites right now?
I didn't know we had great whites here. So.
Yeah, I was going to say, like, this is where you are, Blake, and just to let you know. You're safe.
It's funny you say that. I have family in southern Georgia, and I'm from Atlanta originally.
Oh, are you?
I'm actually not safe. There's war there.
Yeah, well, just know that this is for now. If you see those red dots coming onto land, I don't know about what'd you say?
Sharknado.
We got Sharknado. Great whites falling from the sky. I'm worried that they've evolved legs. Somebody else has said something bigger has eaten them, like Godzilla, and is now moving across land. So all I can say is, if they come towards you, this is when it's good to live in Arizona. We can arm ourselves against the great whites. So there's all sorts of other fun stuff. So the phenotyping aspects.
Gil could just come through a little bit better.
Wait, what?
If the recent Hurricane Gil or how.
Oh, yeah.
Yeah, really.
It had come through a little bit better.
We were this close. We were this close to having sharks drop from the sky. But no, it went and dropped them on New Mexico instead. And like many people here, I gave my group the day off and said, work from home, it's going to be chaos. No sharks, no rain. But we do have phenotyping. We do have high throughput phenotyping. And so what I want to show is this is a really neat setup by a group up at the University of Wisconsin, Edgar Spalding's group. And what he does is he sets up these cameras, and what they're doing is they're focusing in on these petri dishes where he's scattered out Arabidopsis seeds or seeds of other plants. And he's making movies, time-lapse movies, of seeds germinating. So he gets to watch something that we actually don't know much about, which is the dynamics of root growth, as well as the cotyledons that are coming up. And he generates a really good amount of data, I mean, relatively speaking, for a relatively small amount of money. This is the kind of information that gets fed into iPlant's image processing platform known as BISC that Nicole was talking about, and it's just phenomenal in terms of what we're learning about these dynamic processes. In other words, we at iPlant have high tech to help you watch grass grow. And we can do this on a larger scale too. So this is actually a really impressive piece of equipment up at the Maricopa Agricultural Center outside of Phoenix. Has anyone been up to the Mac Center? Oh, you should get up there. It's cool. It's actually pretty cool. Big USDA research facility. A lot of folks from the University of Arizona are involved out there as well. And what they have is this highly modified, well, so this is a standard, what they call a high tractor, I think, one that can drive over a lot of crops. And then what they've done to it is they've gone through and they've affixed to it a whole bunch of different kinds of equipment that allow them to measure plant height with LiDAR. They get a variety of temperature and color with a variety of spectral stuff, GPS equipment. And what they're doing is they're using this as a mobile, high-throughput phenotyping platform to basically watch crops grow. And you can imagine now taking this kind of technology, scaling it to drones, from drones to aircraft, aircraft to satellites, and we have some really impressive ways that we can start to view our planet. And again, huge amounts of data come off of these kinds of projects. So the one thing that I want all of you to keep in mind as you think about data is that transforming data into knowledge only occurs in one place, and that is usually in the trained brain of a scientist. And what we do is we use a variety of computational platforms to help re-represent these data into some form that's easier to feed back into that brain, so that some scientists, again, man, I'm going to use the letter H. Hagen can come take a look at this E. coli sequence, take a look at this representation of it, and say, you know what? These colored arrows are representing genes. This green one right here labeled IS5, that's a transposable element. These are the canonical selfish gene. They make copies of themselves. They hop around the genome, and boom, it's jumped in, landed, and disrupted this other gene called WBBL. And that's what's giving rise to some particularly unique phenotype of this E. coli strain that he has studied for years about. And this is so much of the process of what we're trying to do. And when it comes to big data, what we need to really start thinking about are all the things that are involved with data. When you start dealing with, let's say, a 1 meg file, how do you deal with a 1 meg file if you want to share it with a collaborator? Send an email. Perfect. That starts to fall down when you reach, let's say, gigadata. How do you share gigadata?
Well, the old way, you send a floppy disk to them. Yeah.
With a gig?
Or, I mean, yeah, one of those zip drives.
You do zip drives, you can send CDs, you can send a flash drive. That even starts to break down as you get up into the terabytes, tens of terabytes, hundreds of terabytes. And as you start to think about everything that it takes to move those data around, you need to also take into account everything else that has to happen with data. So it's not just movement of data, storage of data, the analysis of data, visualization of data, the markup of data with metadata. Anyone here not heard of metadata? This looks like a pretty data-savvy crowd. Dave, I'm going to pick on you. I'm picking you too much, yeah. What's metadata?
Data about data.
Oh, perfect. Data about data. Metadata, one of the most important things you should be thinking about as you are going to all become great stewards of data, which is, for example, when it comes to this genome, primary data is the actual sequence, metadata is everything used to describe that sequence. The organism, where it was isolated, where it was sequenced, the technology that went into prepping the DNA, the assembly program, what happened to it.